Right. Good afternoon, everyone. Thank you for joining us for today's webinar. I'm Cassie, Marketing Supervisor at GenScript, and I'll be the moderator for the webinar today. A little background about GenScript for those who are new to us. GenScript is the leading life science research and application service and product provider in various fields from basic life science research to translational biomedical development, industrial synthetic products, and cell therapeutic solutions. And GenScript ProBio is the biologics CDMO segment of GenScript Biotech, proactively providing end-to-end service from drug discovery to commercialization with proactive strategies, professional solutions, and efficient processes in antibody drug and gene and cell therapy to accelerate drug development for customers. In today's webinar, we have Dr. Ying Li, our Director of Downstream Process Development from GenScript ProBio, to present on taking the accelerated path upstream with IND-compliant CHOK1 GenS cell line development. Dr. Li is the Director of Process Development for GenScript ProBio. She leads the CMC process development services for protein and antibody and provides technical leadership in technology platform development. She joined the company in 2022, bringing over more than 10 years of biotechnologies experience spanning multiple specialties, including bioseparation, bioproduction, viral clearance, and MSAT. Prior to joining GenScript ProBio, Dr. Li was an R&D manager in Millipore Sigma in the US, leading the development of the company's gene therapy manufacturing platform. Dr. Li obtained her bachelor's degree in chemical engineering from National University of Singapore in 2012 and her PhD in chemical engineering from the Pennsylvania State University in 2017. She's dedicated to innovation in the bioprocessing field and a frequent speaker at international conferences, including ACS and AICHE. This webinar will take around 45 minutes and we'll have about 15 minutes for Q&A session later. You can type in your questions in the chat box and we will answer them after the presentation. If we didn't get to answer your questions during the Q&A session, we will email the answers back to you after this webinar. Do also help us to fill out the short survey that you can see on your screen, and you will stand a chance to win a GenScript gift pack. So now, without further ado, we will let Dr. Li to start her presentation right now. Dr. Li, over to you.
Thank you, Kathy, for your introduction. Hello, everyone. My name is Ying Li. It's my pleasure to present today's webinar, Accelerated Path Upstream with IND-compliant CHOK1 GenS cell line development. In this webinar, you will learn the introduction of GenScript ProBio CDMO business and ProCLD cell line development service, and the introduction of our new cell line development service, CHOK1 with ADCC enhancement. GenScript ProBio aims to create innovation through collaboration. Our vision is to become your most reliable and sustainable biologics partner. Our mission is dedicated to proactively providing end-to-end service from discovery to commercialization with professional solutions and efficient processes to accelerate drug development for customers. Our business includes both antibody and protein drug, as well as cell and gene therapy drugs. GenScript was founded in US in 2002. Over the years, we have expanded our business to a number of areas, including life science services and products to provide one-stop solutions to global research communities, an industrial synthetic products platform known as Bestzyme, an integrated global cell therapy platform known as Legend Biotech, and biologics development and manufacturing CDMO platform known as ProBio. Our GenScript ProBio brand was officially launched in 2020. We are a global CDMO with broad geographic footprint and customer base, spanning from China, APAC, to USA and EU. Our global headquarters is located in Nanjing, China, with facilities enabling biologics discovery, process development, and clinical manufacturing facilities. In addition, we are quickly expanding our manufacturing facilities in Zhenjiang, China, including plasmid virus process development, virus clinical and commercial manufacturing facilities, as well as biologics commercial manufacturing facilities, which is currently under construction. we are further expanding our global manufacturing footprint into the US in New Jersey. We aim to constructing cutting-edge discovery and process development platform. In our biologics discovery platform, we cover a variety of different modalities, including monoclonal antibody, single domain antibody, and bispecific antibody, ranging from lead generation, lead optimization, and testing. Under our antibody and protein CDMO platform, we cover end-to-end CDMO services, ranging from cell line development, CMC development, including process and formulation development, and analytical and QC development, and early stage clinical manufacturing and commercial manufacturing. Our production technologies utilize both conventional fed batch cell culture as well as perfusion-based cell culture for continuous and intensified process. Our GMP manufacturing facility was built with high quality standard to support preclinical and clinical material supply. The facilities met global quality system specified by ICH, FDA, EMA, and NMPA. Our current capacity is 2,600 liters with expectation to grow into 16,000 liters by Q3 2023. Over the years, we have achieved fast-growing global track record. For our antibody drug discovery platform, there has been over 600 plus lead generation projects successfully completed and 350 plus lead optimization projects being completed, with the most advanced project has been marketed. 13 therapeutic antibody licensing deals have been closed, with 5 SMA body strategic partners being established. For our CMC and manufacturing platform, there has been over 300 cell line development projects being completed, and over 51 CMC and CMO projects being completed. The most advanced project has been delivered in clinical phase 3, with 35% among all the CMC projects originated from United States, Europe, and Asian Pacific. So far, we have helped our clients to achieve 15 global IND approvals and delivered 56 GMP batches. Among all the 32 customer audits, there have been 0 critical and major matters being reported. Next, I will walk you through our ProCLD cell line development service in details. Cell line development plays an essential role in drug development. Creating a robust, high-yielding, and stable cell line is the foundation of any reliable biomanufacturing process. Here shows a timeline for stable cell line development process. It typically starts with a cell pool development process. In general, we recommend the cell pool development process start in late discovery stage in order to generate sufficient materials with consistent quality for animal studies and developability assessment. It is followed by single-cell clone development, which was selected from screening the best performing cell pool. Afterwards, a PCB or primary cell bank was generated from the top performing clone for reference material generation and future process development. And finally, a master cell bank or working cell bank is generated from PCB for GMP manufacture. Here is a snapshot of our ProCLD platform. It consists of whole cell system, stable expression vector, clone ranking, and screening system. Our whole cell is a CHOK1 wild type obtained from ECACC and has been suspension adapted. It is animal origin free and compatible with chemically defined commercial media. We can grant commercial sublicense of our whole cell into our clients for commercial therapeutic development. Our stable expression vector is called pGenHT 1.0 double gene vector expression system. It utilizes the GenS selection system and was developed and synthesized in-house in GenScript with traceable records, thorough QC, and sequence confirmation. These high expression vectors are built for monoclonal antibodies, bispecific antibodies, and proteins. Last but not least, we have a high throughput and automated platform, which include a variety of different instruments that enables a fast and effective single cell clone selection process. Let's take a closer look at our CHOK1 GenS host cell. It was purchased from ACC with a catalog number, and then afterwards suspensionally adapted to serum-free and commercially chemically defined cell culture medium. A PCB was generated and fully characterized by our external vendor. Afterwards, an MCB was generated from the PCB and fully characterized in bioreliance to fulfill the regulatory requirements of US, EU, and CN. On the right-hand side, it shows a comprehensive items that has been performed during the characterization of our master cell bank. It should be noted that we have submissions to FDA, the drug master file submissions to FDA, which helps to support our IND, NDA, and BLA process. It helps save time for the product review and evaluation and reduce the preparation work for drug registration by using the DMF number. The DMF submission includes both the host cell system as well as the vector expression system. Our ProCLD solutions include 3 different service packages: the pre-CLD cell pool development service, the ProCLD cell line development service, and ProCLD plus cell line development process. In the pre-CLD service package, it includes the cell pool development and drug developability assessment. The timeline starts from 10 weeks and is generally applied in late-stage discovery. The benefits of utilizing the cell pool development package include generation of sufficient materials for animal studies, and we guarantee greater than of 1 grams per liter of titer for monoclonal antibody, evaluate the developability of drug candidates, help identify the possible risks in future process development, and the cell clone can be further developed into a stable cell line. The ProCLD service package include end-to-end cell line development service, as well as PCB and PCB stability study. The timeline ranges from 14 weeks, and it is generally applied for all the preclinical CMC development. It offers fast turnaround time. Again, like I said, from gene synthesis to top 6 clones only takes about 14 weeks, and we get guaranteed titer greater than 3 grams per liter for all antibodies. Guaranteed 60 generation stability and compliant for IND filing requirements. The ProCLD Plus service is built on top of the ProCLD service. In addition to the cell line development offering, we also include the feed and medium selection and using Amber 15 bioreactor evaluation. The whole process takes around 18 weeks and good for all the clinical CMC development. The benefits include fast turnaround time from gene synthesis to top 1 clone in only 18 weeks, and the improved titer up to greater than 5 grams per liter for antibodies. The medium and feed selection has been optimized and so as to the bioreactor performance and deliver the most suitable clone for future CMC studies. Why should you consider the pre-CLD service utilizing the cell pool development? Compared with transient expression system, the whole cell and vector system of stable cell pool are the same as stable cell line used in the CMC study. Therefore, consistent product quality could be achieved between the animal developability studies utilizing the cell pool materials in comparison with the CMC studies. During the developability assessment, 3 extreme conditions will be screened, including high temperature at room temperature, low pH, which represent the worst case for purification process, and freeze and thaw, representing the worst case during shipping and storage. And 3 quality profiles will be compared under the 3 extreme conditions, including the stable purity profile, the charge variant profile, and bioactivity profile. And afterwards, the criteria of the drug candidate selection will be finalized before starting a CMC project. This table shows a developability case study. Among 3 different drug candidates, A, B, and C, the stability under 3 extreme conditions are performed using the cell pool material. And their performance for purity, charge variant, and bioactivities and other quality metrics have been compared, and drug candidate C has been chosen and finally succeeded in the future CMC study. This slide shows the general workflow for the cell line development service. It started with the synthesis of target gene and preparation of target plasmid, which were all performed in-house in GenS script. After that, 4 independent transfections have been performed and seeded into 8 of the 24 well plates to generate 196 total cell pools. The top 6 cell pools will be selected based on the high expression titer, as measured by ELISA. Afterwards, single cell clone will be screened in 10 of the 96 well plates as seeded using wipes. And the monoclonality was confirmed using the cell metric cell image method. The top 30 clones among over 100 single clones will be selected for further fed batch evaluation using ship flask. The top 6 clones expression high titers will be further selected for under 15 mini bioreactor evaluation and median feed selection, which includes 24 in total DOE studies. After that, the top 1 clone will be selected for future CMC study. It should be noted that from gen to top 6 clones will takes only 14 weeks, and from gen to the top 1 clone takes only about 18 weeks to complete. Next, I will go through a couple of key features of our ProCLD service. High productivity. The graph on the left-hand side shows the titers for our monoclonal antibody expression. Among all the projects for MAB, the average titer reached to 4.35 grams per liter, with the highest titer up to 8.7 grams per liter. Over 75% of the projects achieved titer greater than 3 grams per liter. On the right-hand side shows the average titer among all the different modalities, including MAB, bispecific antibody, and recombinant proteins. As you can see from the graph, among all the projects, the average titer for bispecific antibody and recombinant proteins could reach to greater than 2 grams per liter. Excellent cell culture performance. This table summarizes a number of different projects with the cell culture performance. As you can tell that the peak cell density reached greater than 10 million cells per ml, with a population doubling time smaller than 26 hours for all the molecules. Excellent stability. This table summarizes 13 different molecules and their stability study. T1 indicates the starting titer at the beginning of the stability study, and T2 indicates the titer by the end of the stability study. The study period covers over 60 days, and the titer variance should be less than 30% by the end of the stability study. As you can tell from this table, for all the tested molecules, the titer variance is much smaller than 30%. Excellent scalability. On the left side, it shows consistent high titer from multiple different clones. For this particular case study, 6 different clones were obtained from 2 different cell pools. As you can tell, for all the selected top clones, there has been greater than 7 grams per liter of the titer. Excellent scalability from 3 liter to 200 liter scale process. This case study shows that when a 3 liter scale process was scaled up, and 3 of the 200 liter scale process were performed. And you can tell that the cell culture performance, the titer, and the charge variant or other quality metrics are very comparable between the 3 liter and 3 of the 200 liter run. Next, let me briefly introduce our GM Pro cell banking system. The generation of a regulatory compliant cell bank is an essential element in the production of biotherapeutics. Cell banking assures that a uniform population of cells is preserved and their integrity is maintained, and that a sufficient supply of material is readily available for the life of the product. Of equal importance is the maintenance of these banks in a secure, controlled, and monitored storage environment. This slide shows our CGMP compliant cell banking laboratory with different facilities that enables a regulatory compliant cell banking environment. And also we have excellent documentation system which ensures every in and out is recorded. In summary, our delivery package for our ProCLD service include a whole cell systems, consists of our CHOK1 GenS cell line and proprietary PGen HT 1.0 DGV vector, a full traceability documentation system, detailed protocols and guidance, as well as technical support to ensure your success. In conclusion, our ProCLD service does help to help you closer to success. Our platform includes the key technology drivers in our whole cell system, proprietary vectors, and high throughput and automated screening platform. We offer accelerated timeline from gensynthesis to top 6 clones in 14 weeks and to top 1 clones in 18 weeks. High quality delivery, including high quality and productivity prior to cell culture. And for mAb molecules, there is guaranteed greater than 3 grams per liter titer and average 4.35 grams per liter of the titer, and up to 8.7 grams per liter for titer. Excellent stability have been observed in all of the projects in over 60 to 90 generations passage. We offered regulatory compliance with traceable records for host cells and vectors, traceable records for cell line development, and cell image to demonstrate monoclonality. There has been an excellent track record with over 300 cell lines developed, 149 projects for IND filing, and 18 currently under clinical stage. Next, I will give you a brief introduction of our new cell line development service, CHOK1 with ADCC enhancement. ADCC, antibody-dependent cellular cytotoxicity, is a mechanism of cell-mediated immune defense whereby an effector cell of the immune system actively lyses a target cell. ADCC is important in the efficacy of cancer antibodies, but with many approved cancer antibodies, there is less ADCC than could be desired due to non-specific IgG competing with the drugs for binding to Fc gamma receptors on NK cells. It has been found that afucosylation monoclonal antibodies overcome this problem through improved Fc gamma binding. So removing or reducing afucosylation is a good way to enhance ADCC activity and efficacy of cancer antibodies. For our ProBio platform, our CHOK1 ADCC enhanced cell line has been achieved through knockout of the FUT8 gene to produce afucosylated antibodies. The CHOK1 ADCC Plus host system includes a proprietary CHOK1 ADCC Plus cell line system to produce afucosylated antibodies. Transient expression cell line for different customers in late stage discovery or CMC stage. Comprehensive analytical methods for ADCC performance evaluation. And excellent track record cell line development for our whole cell with 1890 approval already achieved. For whole cell system, CHOK1 cell license could be chosen from with either and type CHOK1 GenS and ADCC enhanced CHOK1 accumulated price when purchasing combination of the two systems. The system is royalty-free, and we offer direct sub-license from ProBio to our clients for therapeutics development. There are a couple of highlights for our ADCC enhanced cell line. In terms of productivity, as shown in this case study, for the fed batch culture, the titer for CHOK1 ADCC Plus cell line is very comparable to the wild-type CHOK1 GenS in both of the 24 deep well plates as well as 3-liter bioreactor. The CHOK1 ADCC Plus cell line shows a good performance in cell growth and viability, and is very comparable to the CHOK1 GenS cell line. This slide shows the confirmation for afucosylation modification. This mass spectrum shows that in comparison to the original GenS cell line, the afucosylation site in G0 and G1 has been knocked out in the ADCC enhanced cell line. Therefore, the antibody is successfully afucosylated by knocking out FUT8 gene. This last slide confirms the ADCC activities of the enhanced cell line. The first first graph on the left-hand side shows the affinity to CD16 receptor has been increased by 3 to 4 times in comparison with our original GenS cell line. And the middle graph shows the potency using the reporter gen-based ADCC assay. As you can tell, at the same concentration of antibody, Afucosylation antibody has stronger ADCC activity, which has been increased about 10-fold. The last graph shows the PBMC-based ADCC, which is a cell-based in vitro assay. The cytotoxicity of the ADCC plus cell line has increased by about 10%, and the effect concentration, EC50, has been reduced by 6 times. And overall, the ADCC effect was significantly enhanced. This brings to the end of today's webinar, and thanks for listening, and look forward to hearing any questions you may have. Thank you.
Thank you, Dr. Lee, for the great presentation. We have a few questions from the floor, so let's just answer it one by one. The first question, what are the key aspects during the cell line development of asymmetric bispecific antibodies?
Yeah, this is a good question. It's not specifically covered in this webinar. During the structure of asymmetric bispecific antibodies, we typically utilize multiple plasmids to express the 2 different chains in the bispecific antibody. Therefore, in comparison to the monoclonal antibody, which we only utilize one double gene expression vector, we utilize multiple plasmids and optimize the ratio of different plasmids in order to ensure the productivity and efficacy of the expressed bispecific antibody, and making sure that assembly is effective.
All right, thanks, Dr. Lee. The next question from the floor, how to evaluate and select clones with enhanced ADCC effect using FUT8 KO cell line?
Yep. So for our ADCC enhanced cell line, the selection system is still based on the GenS system. And after the stability of the clone were evaluated, the selection process utilized the assays as has been presented previously. So we utilize the mass spectrum to confirm the Afucosylation, as well as using a variety of different assays to confirm the ADCC activities. Therefore, during the single clone selection process, we utilize both of these assays to ensure that the selected clones could express enhanced ADCC activity. Thanks for the question.
Thanks, Dr. Lee. Yeah, no problem. The next question, what are the indicators are generally examined in the drug development assessment?
Yeah, I think we briefly covered this during the presentation. Let me browse the slides. So in this slide, we actually covered the different conditions being evaluated during the drug developability assessment. So briefly, in order to select the candidates with the best stability, we have screened 3 extreme conditions under high temperature, low pH, and freeze and thaw. And 3 quality profiles were compared under the different extreme conditions in order to help select the best performing drug candidate. this is essentially the process for a typical drug developability assessment that happened in early stage or late stage discovery process.
All right, thank you, Dr. Lee. The next question, for complete MAB clone development, will you use heavy chain and light chain separately or in the same vector?
Yeah, I think we briefly covered that in our first question. So it depends on the most effective assembly of the bispecific antibody. Again, if we find expression, both heavy chain and light chain on the same vector gives the best performance, it could be utilized using the same DGV vector. If we perform optimization study and shows that different ratios of plasmids would help enhance the assembling efficiency, we can potentially choose to express the chains in different vectors.
All right, thank you, Dr. Lee. So for all the attendees online, if you have any questions, please feel free to key in your questions in the chat box. And if there's no further questions, right, I think we can end the presentation a bit early. So for those questions that will come in after the webinar, we will still email you the answers from Dr. Lee back to you. So thank you, Dr. Lee, once again for the great presentation, and to all of us who joined us today. I hope you find this session beneficial for you. Oh, there's another question. Maybe we can take the questions before we end off the session.
Yes.
Yeah. So this question, it will be, can you explain a bit about the license issue, like for your vector and also the host?
Yes. I think from my understanding, we can directly sub-license our expression system, including the host, as well as the expression vectors to our clients. And for further details, you can reach out to our business development team for details.
Okay, I hope that answer shortcut your questions. Yeah. So if there's no further questions, I think we will end off the session for today. Thank you everyone for joining, and thank you, Dr. Lee, for the presentation.
Thank you.
All right, thank you everyone. Bye-bye.